Okay, let's get started then. Hi everyone, and welcome to the first workshop of the AaRCademy series organized by the Animal Ancient DNA Research Community. A lot of you are maybe part of our community, have heard of us, or if you haven't, you can find more information of us on our webpage shown here. We're also active on Blue Sky and also on the Elements platform, which we highly recommend those of you who haven't joined to join because it's a nice way to kind of for people to ask questions regarding analysis of their own data. Different preprints and papers are shared, job positions are posted, and events like this workshop series, but also other conferences and seminars are also posted as well. And the occasional cute animal photo will be posted as well. So you can find instructions on how to join on our webpage, and if you have any questions, feel free to shout out on how to join it. So, focusing on today's workshop, we are really lucky to have Nikolay Oskolkov from, well, this affiliation should change now, but who was previously at Lund University and will be talking to us about ancient DNA data processing. A brief intro about Nikolay is that he did his PhD in theoretical physics and then moved on to computational biology, if I'm correct, at University, with an initial focus on medical genetics. And then in 2016, he became a senior bioinformatician in the SciLife group in Lund as well. And he's worked on various topics around molecular biology and evolutionary biology, going from transcriptomics to multiomics, single cell analysis, metagenomics, and population genetics, and has also developed very important tools widely used in ancient DNA studies with a focus on ancient metagenomics. So we're really lucky to have him here today, and he has also started his own research group now at the Leos Institute in Riga, Latvia, where he currently is. And excited to see him starting his own group. So if you have any questions during the workshop, please raise your hand first in Zoom, and then you can unmute yourself. But generally, please keep your microphones muted unless you're asking a question. Before we start, I would briefly like to advertise the next workshop of our series, which will take place on February 3rd, again online. And Pablo Librado Sanz will be talking about different methods for asking questions about demographic history. So with that, I'll stop sharing my screen and give the stage over to Nikolay.
Thank you, Katya. Yes, hello everyone. While I'm sharing my screen, I have to say that I really admire what AaRC community is doing. So thank you very much for doing this. I think it's very important, and I occasionally join your seminars, and I always find them so useful, so informative. So please keep doing what you're doing, and I really appreciate. And I think many people appreciate as well. Hello everyone again, and I'm really happy to greet all of you. Greetings from Riga, and yeah, I'll tell you a little bit more about my background. But I'm very happy to meet all of you virtually, guys. Yeah, so I'm going to talk about ancient DNA data pre-processing or processing, and I slightly changed the title. I hope the organizers will excuse me. I added this a metagenomic perspective because I'm really I have my background in genomics, so I did lots of genomics, mostly human genomics, but I was doing a lot of biomedical research, but I really like this metagenomic view of doing analysis, and I'm going to explain why it is useful and why I'm promoting this metagenomic perspective. So let me, yes, by the way, before we continue, so we are not so many, so I encourage you, so if you have questions, so it's going to be 2 hours seminar or workshop. And if you have, burning questions, I have no problems if you just raise your hand and unmute yourself and ask. I don't know if the organizers are okay with this format. I don't mind if you just jump in and ask a quick question so that otherwise I will continue talking, and maybe you don't want to keep your question until the end, so it's a good idea just to ask when you have this question. Yeah, so let me briefly introduce myself. So I'm originally coming from theoretical physics. So I did my PhD in theoretical physics in Moscow. And then in 2011, I met medical doctors from Lund University in Sweden. So it's this beautiful building of Lund University. And they convinced me to transition from physics to life sciences. And this is how I started doing genomics, working at Lund University, doing type 2 diabetes research. So I have my background mostly in genomics of type 2 diabetes. And then in 2016, I was recruited by Scilab Lab. It's sequencing facilities in Sweden. So it's kind of life science infrastructure in Sweden. And for many years, for 10 years, I belonged to the bioinformatics platform of Scilab Lab that is called NBIS, the National Bioinformatics Infrastructure Sweden. And during my career, I was in bioinformatics. I was I was involved in very diverse projects, varying from cell biology to image analysis, metagenomics, a lot of statistical analysis, a lot of machine learning, a lot of biomedical data mining, integration. And also, I published quite a few papers in ancient DNA and mostly in ancient metagenomics. That's my kind of specialty. And this is what mostly the data processing steps that I'm going to promote today. They're coming, they're inspired by this microbial analysis, and I believe people from from classical genomics, from working with single organism, they might find what I'm suggesting interesting, and I hope it will be useful for you guys. So currently, I'm actually transitioning from Lund University to Riga. So I'm starting my own group in Riga. So there is a funding for 5 years, and there are postdoctoral fellow positions and PhD student positions open in my group. So please contact me if you're interested in working with me. So it's the group is called the Metabolic Research Group. And it's about machine learning method development for biomedical applications, mostly, but not only. So it's a capacity building project. So we are doing lots of different types of analysis, including genomics. So I think it's quite relevant for this audience. So it's currently only me and Daniel Rivas so far. So we are very happy to have Daniel Rivas on board, who's an excellent expert in AI and machine learning. So we are working currently in this metabolic research group, and we are waiting for one more postdoctoral fellow, and there will be 2 PhD student positions open this fall. So please, if you are interested in working with me, please let me know. So my expertise in ancient DNA mostly comes from 2 milestone papers. So the first paper, it's a method paper called aMETA. It's a workflow, but I rather call it a method because it's not like a classical pipeline, but it's more like an approach. And that's why I always advocate that aMETA is not just a pipeline; it's kind of an approach. And you can if you use the same mentality as aMETA uses, you can do your own pipeline or run a pipeline similar to aMETA. So aMETA may be not like the best computationally friendly and easy to run, but certainly the approach, the principles of aMETA, I'm quite certain that they're I believe in them, and I think they should be used in ancient DNA analysis as much as possible. And so this method can be installed from the GitHub page, and this is a great team behind aMETA. So I was developing aMETA in collaboration with my colleagues from the National Bioinformatics Infrastructure Sweden, and also in collaboration with the groups of Anders Gurreström and Louis Dahlén from the Center for Paleogenetics in Stockholm. So I acknowledge all my great collaborators. And the second paper that I'm going to cover quite a bit during this presentation. So it was published in GigaScience just a few days ago. So it's another kind of important study for me, at least. And this method, what we call GenX, so we kind of developed another tool called GenX, genomic exogenous sequence detection. And the project was done in collaboration with Tom van der Valk, the young DDLS fellow, PI, also from the Center for Paleogenetics in Stockholm, and also Pete Heinzmann, also from the Center for Paleogenetics.
Nikolay, could you minimize the floaty from Zoom of different because it's covering the slide. We can't.
All
Well,
right.
it's currently it's covering Tom and Peter's faces.
So maybe
And
you mean like this?
so the control, the annotations, and also the with the each per like the yeah. So now that's good, and also the
Better.
I can't speak the videos of the participants on the side is also covering the right part of
Right. Yeah,
yeah
so I'm a little bit
yeah.
bad in the Zoom settings. I actually don't know how to do
Maybe
it.
the one on the left, the first more
This one.
yeah.
Yeah, is it better? Okay,
Awesome.
excellent.
Thanks.
Thank you, Katya. Thank you. Yes, correct. Otherwise, it was covering quite a lot of the slide. Thank you. And yeah, so when I was planning this seminar, I thought so there were different suggestions, and I thought maybe to run, some live coding session is a little bit too ambitious because usually, just based on my experience, this is something non-trivial. Yeah, so something goes wrong usually, and also yeah, it becomes slow, and you guys probably don't want to sit and listen to my these variables should be called in this way, and yeah, better concentrate here on the concepts, on kind of science, and I explain the ideas, and I hope it will still be useful and informative for you, even though less hands-on. So if you join this seminar and expected more hands-on, probably yeah, that was a little bit too ambitious. But I will I certainly can if you have technical questions, I certainly would be happy to answer them. So otherwise, the material that I'm going to use during this presentation, it was covered to a large extent in this Faisal Ancient Metagenomics course that I was running like a few months ago. And it's a very comprehensive material with slides, with videos, with notebooks. And this is free, it's publicly available. So if you're interested in covering some of these methods and ideas that I'm going to talk about now, you're very welcome to just clone this repository and use this material. Yes. So let me now start with some introduction. And I said that we are going to use this metagenomic approach to ancient DNA data analysis. And it's a little bit maybe I don't know. My personal experience is that not everyone kind of understands this approach, and not everyone is a fan of this approach. But let's see whether you like it. So I'm going to explain what I mean by this metagenomic approach. So I'm basically suggesting to see any sample as a metagenomic sample, unless you are sure that there is like maybe a single organism present there, and unless you have evidence that it's not a metagenomic, it's a single genome sample. But let's take it slowly, step by step, and I hope you'll find it interesting. So let's say you have, let's take a classical, it's an animal ancient DNA community here mostly, and I believe that's your like a typical setup. So you have a bone or tooth, maybe it's a mammoth tusk. Let's say we are working with a mammoth tusk. So we are going to go to their this clean lab, ancient DNA facility lab, and drill, extract DNA and sequence it. That's not my competence, unfortunately. I myself never did it to my, yeah, it's my tragedy, basically, but I still, I don't know how people do it, but this is what I kind of observed and what I read. But I'm a bioinformatician, so this is my data. So usually we computational people, data analysts, we get the data from the lab in the form of short nucleotide sequences, which are called reads. And if it was a mammoth task, maybe you would expect that this will be mostly mammoth DNA, mammoth reads. However, are you sure? Is it guaranteed that all these reads are actually coming from a single organism, which is a mammoth? Well, at least it's quite plausible that we human, when we do the sequencing, we can drop a little bit of our DNA, or even not on the sequencing step, but maybe sample collection and so on. So it's quite it makes sense to assume that there can be some human contamination. Some of the reads may very well originate from humans. And also, if you're a good analyst, you may suspect that there can be, more than just two organisms, mammoth and human, but maybe there can be some bacteria or microbial DNA present. So this is not impossible to assume that there are more than just one organism present. And this is basically, you start thinking in a different way. You start thinking in this metagenomic way. So if you assume that there can be more than just single organism, a DNA from more than just single organism present in your sample, that changes quite a lot the way you're doing analysis. So if you ignore this fact that there can be more than one organism present in your sample, how would you start? Well, you can do this traditional step. You take this reads from the mammoth task and you align them to an elephant reference genome as a proxy for mammoth reference. But then, so if you want to be a little bit more open-minded, you assume that these reads, they not all of them might be mammoth reads. Some of them can originate from humans, as we just suggested here on the previous slide. So some of these reads can be green. And what to do, how to handle this problem, how to disentangle, how to figure out what reads are coming from the mammoth and what reads are human reads. So you can do this approach, which is called competitive mapping. And this is a very, very straightforward, very elegant approach that I highly recommend. Within this approach, you would like to actually concatenate your elephant reference genome with a human reference genome and align your collection of the reads to two genomes simultaneously. And it is called, this mapping is called competitive because all your reads are going to compete, or sorry, all your reference genomes are going to compete for the reads. So basically the reads have to decide what is a high affinity. Do they have a high affinity to the elephant reference genome, or do they have a high affinity to the human reference genome? In this way, you can disentangle, or there is a potential at least to disentangle the human reads from the elephant reads. And of course, there can be mismappings still. Some of the human reads will remain. Many of the reads will become ambiguous, so they will map with exactly the same affinity to both reference genomes, but probably in this case you would like to drop them because of this ambiguity. So it's hard to say. You can never be sure that these reads, mapping with the same affinity to human reference and elephant reference. You can never be sure that they're informative. Actually, they can be. So it's risky to keep them in this elephant reference alignments because they might be human and they can distort your downstream computational analysis. However, if you sort of what I suggested here on the previous slide, if you want to be even more open-minded, you can say that, okay, it's quite plausible that lots of these reads, they're neither elephant nor human, but they're microbial. And the way you would handle this, you would actually like to concatenate your elephant reference genome with your human reference genome and with a bunch of microbial references. And of course, there can be, thousands and tens of thousands microbial reference genomes. And a good thing about microbial references is that they are small, so they're not that difficult to handle computationally. But otherwise, this ultra competitive mapping, when you concatenate several reference genomes, this gradually kind of clean your organism of interest or the alignments to your organism of interest much more accurately. So basically, this competitive mapping is, in my opinion, it's a very central part of our aDNA analysis, and I'm advocating to use this competitive mapping approach as much as possible. I know that not everyone thinks in this way. People who are coming from more population genomics background, they rarely do it in this complicated way. They're usually, like my personal experience, I don't know, maybe I'm wrong, but my personal experience is that people who work with a mammoth task, they usually align their reads to just elephant and maybe elephant plus human, but not really to elephant plus human to and plus microbial. That's a little bit too much. And I personally think that the more organisms you add to this database. So this is what in metagenomics it's called a database. So the more organisms you have in the database, the better. So people in metagenomics, they never assume a single organism. They always think that there can be thousands, tens of thousands, and even millions of organisms present. So if you think that this approach is too complicated and maybe strange, I would say that if you assume that there is only one organism present in your sample, it can be a little bit risky. So it's, of course, you can do it at your own risk, but there are many examples actually when it can go very badly. So here I'm just giving you one example of a Nature paper retracted, and this is kind of not an ancient DNA example, and it's not really about this, mammoth DNA at all, but it's a mixing and confusing different organisms in the same sample. So the authors of this paper, they thought that they were working with microbial DNA from TCGA project. It was a cancer project. So the aim of this paper was to analyze microbial DNA from cancer patients, but it turned out that there was a bunch of human DNA involved into this analysis, and finally, the paper was retracted. It's a kind of it shows basically that it's very non-trivial to disentangle different types of DNA, and if you concentrate only on one organism and you assume that there is only one organism, that can be risky. So I really encourage everyone to think in metagenomic way. So basically to approach this sample and say, okay, I know that it's a mammoth task, but am I 100% sure? Am I confident that it's going to be only mammoth DNA? How confident I am? And in this metagenomic approach, there are a few more methods popular other than alignment. I would say in genomics and single organism genomics, it's mostly alignment, maybe de novo assembly, what people are doing. In metagenomics, there are different approaches. So in addition to the alignment approach with BWA or bow tie, people often run these k-mer-based classifiers, such as Kraken, Metaflan, and so on. So there are ways to skip this alignment step and quickly get some understanding what is inside your sample. But by doing this k-mer-based quick lookup, using some lookup table to quickly check the composition of your sample. At least in this way, you can be more sure that you're working, with just single organism. Why not to do it? Just to quickly, as a sanity check, as a quality control, to check what kind of organisms are present in your sample. And like to my experience, you will be surprised to see that there is more than one organism in your sample. That's my expectation, at least my prediction. But of course, you're very welcome to try it. And this alignment and classification approach, they belong to a reference-based approach. Here, there is a strong assumption that your query reads they have a high similarity to the reference, and this might not be fully, fulfilled. This assumption might be violated depending on the divergence between your reference and your query reads, biological divergence. It can be technical divergence just because it's a degraded DNA. So maybe the reference-based approach is kind of has own disadvantages because it has this strong assumption, similarity to the reference. De novo approach is, in theory, is kind of unbiased, but it has own challenges. So I wouldn't say it's easier to use de novo assembly, especially for ancient DNA people with very short reads, with very low coverage samples. So I would say de novo assembly is certainly not easier, even though in theory it's more promising because it doesn't assume this similarity to the reference. But computationally, it can be a headache. Yes, so now, so once I described, explained this overall my mentality, so I explained what this metagenomic approach means. So I'm going to very briefly go through a few steps in ancient DNA. And just because, just to make sure that everyone is on the same page, and I understand that here in this community where people talk a lot ancient DNA, many of these steps will be trivial, and that's why I will try to concentrate on lesser known things. So I'm going to kind of go through the same steps that are typical in ancient DNA genomics projects and even modern genomics projects. But I'll try to maybe give you a few insights that are lesser known things, and hope you'll find them useful. So ancient DNA, it's a sort of DNA from archaeological samples. To my naive understanding, some people may disagree with this definition. But for me, as a data analyst, I think so. I'm thinking about these limitations of ancient DNA compared to, for example, medical genomics. In ancient DNA, we are working with very little DNA, and it's very hard to do any statistical analysis. So compared to modern genomics and biomedical genomics, where I previously had experience, it's a dramatic difference in terms of how difficult it is to make statements or claims in ancient DNA. So lots of things, interesting things that you want to, lots of inferences that you can do from ancient DNA. They're based on very, very low statistical evidence. That's why the level of uncertainty is huge in ancient DNA. And maybe when people, there are beautiful models in this area which I admire. I think people are putting a lot of intelligence, developing beautiful methods and model, and making this astonishing inferences about maybe migrations, human migrations back in time. But I'm always kind of thinking about these potential artifacts coming from this very low statistical evidence. So the ideas are beautiful, but do we have enough data to be confident about our statements? And moreover, we often cannot validate them. So the validation is what is kind of I think that one of the major bottlenecks in ancient DNA analysis, because we can't go back in time and check in that particular population how their genetics looked like, and people just in ancient DNA say, well, we can't validate it. We can only predict how people migrated. But it's a little bit, if you're used to this machine learning mentality in medicine, it's a little bit strange that you cannot validate, because in terms of machine learning, so if you can't validate your model, you shouldn't predict. And in ancient DNA, it's quite common that we predict. We kind of make this inferences about demography, for example. How was the population size, or effective population size back in time? But there is no way we can validate it. And it's a little bit strange, because if you can't validate, you shouldn't even do these statements, at least from the machine learning perspective. That's my personal kind of difficulty with lots of analysis in ancient DNA. As you probably the majority of you know, ancient DNA is very degraded. It's very short. It's very short. So that's actually the problem with short reads is not that it's difficult to align them. The main problem is the specificity. So basically, you can never be sure that ultra short reads belong to the organism you are thinking about. That's a problem. Yes, you got a mammoth tusk and you got a bunch of reads, but since they're short, can you really prove that they're mammoth reads? And here there is a problem. If it's if it's a 30 nucleotide base pairs reads, they can very well be microbial, for example. There is a very little you can do to actually prove reliably that this ultra short read is actually mammoth read and not microbial read. Because on this scale, on this tiny scale, microbes and mammals can be identical. Yeah, so we need longer reads just to ensure the specificity. Again, we're using this metagenomic mentality, assuming that there is more than just one organism in our sample. Of course, if you assume that there is only mammoth DNA, then you're lucky. But if you assume that there can be another organism present, then you might want to think that these short reads, they actually do not allow us to confidently say what kind of organism is there, because they're lacking specificity. So there is this domination, strange chemistry going on when you get this artificial mutation, transition mutations, and this actually what people realized a long time ago, that this is a gift. That this domination, yes, it makes pop-gen analysis difficult because it gives you some non-germline mutations, so to say, and complicates population genomics analysis. On the other hand, it gives you a very unique way to authenticate your reads. So people found out a long time ago that this increased frequency of transition mutations at the ends of the reads. That's a unique way and very reliable way to actually disentangle modern reads and ancient reads. And nowadays, I think it's quite popular in pop-gen analysis, especially in ancient DNA pop-gen analysis, to eliminate this domination in the lab, doing UDG treatment or user treatment. And there are different opinions here. My opinion is that you shouldn't, eliminate this valuable, unique signal in the lab. But most of the people in the field probably will disagree with me. I would rather fix these problems, PopGen inference biases due to deamination. I would rather fix it bioinformatically instead of manipulating with the data and eliminating this valuable deamination signal in the lab. In my opinion, that's a little bit of manipulation with a very unique a gift signal coming from the past. Yes, and the ancient DNA pipelines, they kind of involve similar steps to modern genomics pipelines. It's again this quality control, trimming, alignment, and genotype calling, and so on. There are some peculiarities. So basically, all of these steps they're a little bit different for ancient DNA. You probably have this unique merging step where overlapping reads pair. If you do pair-end sequencing, then because your DNA fragments are very short, sometimes this read one and read two, they can be merged together. So that's quite unique. People don't do it with modern DNA. And also other steps such as damage. Well, damage doesn't exist for modern DNA analysis. Although fragmentation, I would say fragmentation is not uncommon in modern DNA. Well, cell-free DNA is fragmented, for example. It can be modern, but cell-free DNA is fragmented. So in this way, just the fragmentation, I think it's not enough to say that you're dealing with ancient DNA. The fragmentation means fragmentation. It can be not only due to this power of time, but it can be due to different reasons, like preservation, how you handle your samples. This can lead to fragmentation and so on. So basically, I'm going to go through the steps one by one. Some of them I will cover very briefly. Of course, I understand that time limitation for this seminar does not allow kind of to give you a comprehensive picture of each of the steps. And I apologize if you're new to the field. Of course, maybe you will not learn, everything from scratch in this seminar. But I believe many people here are quite advanced in ancient DNA analysis, and for these people, I will try to give, as I said, a little bit lesser known things to share my experience, maybe. And I hope you will have questions. We can have a nice discussion. Otherwise, all these typical approaches for ancient DNA analysis, they're described in a few papers. So I highly recommend this paper from Ludovic Orlando, like a primer paper explaining the principles of aDNA analysis, where all the steps are explained in much more details. And there are a few more papers that are good to read. So I'm going to share the slides. So I hope you can, if you're not familiar with these papers, you can check them later. So I will start with just all these steps here in this pipeline, including quality control. But I'm not going to teach FastQC. I remember times when I started doing bioinformatics in 2011 or 2010, I think. At that time, FastQC was like a top-notch tool, and not so many people knew how it worked. Nowadays, I think bioinformatic competence increased dramatically. Like nowadays, I actually rarely see people who cannot do bioinformatics at all. Nowadays, like people even working in the lab, they can do R and Python and BWA, and it was not the case like many years ago. At that time, indeed, like bioinformatics competence was a little bit special, but not anymore. I think even people who don't do bioinformatics, they must have heard about FastQC. That's why again, I'll try to give you something that you might not know, I assume. And also, I will emphasize this, how it is useful to use this metagenomic mentality to approach aDNA projects. So when you're looking at FastQC stats, what is very informative for me is to look at this GC content, for example. And if you have just one organism, this distribution will be unimodal. But if you see like two peaks, that's already on the QC step. You can suspect that there is more than just one organism present. So that's very important. That's one statistic that I always pay attention to. And of course, the adapters you have to check that they're present, and you have to actually check this before adapter removal and after adapter removal. You learn a lot just looking at the stats. So I think it's pretty much standard that in all pipelines you actually check you do this quality control before adapter removal and after adapter removal and see how all the stats they change after adapter removal. So for example, if you detected adapters and your reads before adapter removal, they all of the same lengths. So there is no kind of distribution of read lengths. So read lengths is of course it's one of very important stats for ancient DNA people. That's a demonstration of DNA fragmentation that they always watch for. Yeah, like before adapter removal, this all reads the same lengths, but after adapter removal, you get this nice distribution, and you can estimate how degraded your DNA. What is the mode of this distribution? So typically it's around 30, 40, 50, maybe not 100. 100, it's closer to like modern DNA. And but also when you do this merging of overlapping reads. So this tail of the distribution can very well extend to 200 maybe, because you can assume if you overlap two reads of maybe 100 read lengths, then the merged thing can be very easily up to 200 and so on. So here in this case there was no merging. That's why here you stop at 125. So after and so this is how you doing this QC. You detect adapter presence, and there is another funny example how you can judge whether adapters are present or absent in your particular sample. So there is a nice illustration of this metagenomic mentality related to adapter removal. And I call this example the carp in the soil, because you typically see it in metagenomics project. Of course, you will not see this interesting phenomenon if you are working with single organism genomics. But in one of my metagenomic projects, well, not only one, but in quite a few, sometimes when I was profiling my sample and just asking general question, what kind of organisms are there? Let's pretend that I don't know that this was a mammoth tusk. Let's pretend I don't know this. Let's do just quick profiling in metagenomic fashion and see what kind of organisms I detect. And often, well, not often, but sometimes I detected this strange organism, Cyprinus carpio, which is a fish. So how come can you get, a fish? Well, in this particular sample, it was a soil sample, so maybe well, you wouldn't expect to find a mammoth there, but not always. But fish, why not? In the soil to find fish DNA, why not? And then, of course, you can make this beautiful stories. You start interpreting, okay, maybe this soil is coming from a place. Well, to the best of my knowledge, it was not like a lake or anything like this, but maybe there was a lake, back in time. That's an interesting, interesting hypothesis. Unfortunately, that just means that you forgot to trim your adapters, or your adapter removal didn't work. So this is how it's this funny example is actually related to adapter removal. So it turns out that the reference genome of Cyprinus carpio, common carp, was assembled on adapters until 2021. I think nowadays they changed it in the NCBI RefSec, but previously, before 2021, I think the Cyprinus carpio reference genome was full of adapters. And if you, for some reason, did not trim your adapters or forgot to trim your adapters, or you think that your pipeline actually includes adapter removal, but it didn't work for some reason, then you could easily find this strange organism, which was an indication for me. So later I learned that if you see this organism, it means something didn't work with your adapter removal. And of course, if you're running like single organism approach, like here, you maybe assume that there is, let's go quickly back and say, okay, maybe here you think there is only mammoth DNA because it was a mammoth task. It would be strange to actually concatenate your elephant reference with a Cyprinus carpio reference. But if you're that open-minded, and if you happen to concatenate your, organism of interest, the reference genome from your organism of interest, with, a few more reference genomes, including Cyprinus carpium, then potentially it could be useful because you actually will see that if your adapter removal didn't work, then you will kind of discover lots of reads mapping to the Cyprinus carpa, which it's basically a very peculiar, interesting, funny way to see how badly this adapter thing can propagate to all types of your analysis. Otherwise, so there are different tools developed for adapter removal, and I think I worked with all of them more or less. There are benchmarking papers comparing all of them. I would recommend this paper from Gabriele Renaud, like one of the latest ones. But there are actually quite a few papers, benchmarking papers, and my take-home message would be that they all are good, actually. In terms of the error, I think they all are comparable in my eyes. At least this was my understanding from all this benchmarking efforts. But I typically use these two guys, the CutAdapt and FastP. CutAdapt I use for its simplicity, but because I totally understand this tool, but unfortunately, CutAdapt cannot merge overlapping reads, and that's why this FastP is probably the champion. That's a tool I would recommend for both modern genomics people and modern metagenomics, and also ancient genomics and metagenomics people. There are two nice things about FastP. First of all, as I said, it can merge this overlapping parent reads. The second thing is that if you happen to work with Novasec and NextSec technologies, it turns out that this is like two color technologies, and there is kind of a natural kind of over representation of this gene nucleotide. It's again when you check the FastQC quality control metrics, then you would see this particular increase of the gene nucleotide. And this is probably, so it has something to do with this two color technology in contrast to four color technologies such as HiSeq. So this two color technology was a little bit blind, and like the nucleotides that could not be called reliably were actually called as G. And that's why you have this artificial increase of G nucleotide, what's called poly-G tails. And FastP actually has this nice default mode. So by default, FastP detects these poly-G tails and removes them. And this is how it looks like after removal of poly-G tails. So that's why I highly recommend FastP, just for these two reasons. The merging, I mean, it's relevant for aDNA people, especially, so the merging of overlapping reads and, this automatic detection of poly-G tails. I think CutAdapt and maybe other tools, they can still detect this poly-G tails, but I'm pretty sure in CutAdapt it's not by default. I think you have to add some special flag, but FastP does a lot of things by default. Yes, so let's move on to the mapping to the reference, so let's say we check the quality control metrics. Yes, so we did the adapter removal. So what next? So next we are going to continue with mapping to the reference genome, and here again, let's ask ourselves. Do we have just a single organism present or not? How sure are we? Are we completely confident that if it was a mammoth task, that it's going to be like only mammoth reads? Yeah, so if you want to eliminate potential, non-mammoth reads that are likely, contaminants from microbes, for example. Then there is this elegant way proposed in this paper. I think this paper is famous for another thing. So it had something, yeah, it was dealing with this reference bias, which is another issue in genomics and ancient genomics. But one particular method that was, I think, quite elegant proposed in this paper, and also in a Nature paper from 2021, but that it was added only to supplementary the oldest 1 million-year-old DNA from the mammoth. There was another paper from the same group in Nature from 2021 with Tom Walk as a first author. So here is the second author. But I just wanted to, so the papers were about something else, but I just wanted to emphasize this elegant method of which can be used for potentially, eliminating contaminating reads, because if you kind of do the mapping to the reference and check what is the mismatch rate per read for reads with different lengths. So it turns out that for very, very, very short reads, ultra short reads, you will see this increase jump in the mismatch rate. So meaning that kind of shorter reads, they map well, in this particular example, they were mapping with a lot of mismatches. And the null expectation was that this distribution should be flat, that this mismatch rate should not depend like dramatically on the read length. But if you see this can be your potential method for eliminating contaminants. So the reads that map with some increased mismatch rate, those are likely contaminants. And in this paper, I think they did simulations and showing that indeed this jump in the mismatch rate for short reads, it's actually associated with so the reads that demonstrated this jump, they were microbial, they were not endogenous. They were exogenous reads. So it's a nice method, which is not based on competitive mapping. So another way, if you want to disentangle this mixture of different reads from multiple organisms, potentially, I'm not saying that it's guaranteed, but potentially, if you are using this metagenomic mentality, you assume that there is a mixture of reads. This is what I explained on the previous slide. You can go this good old competitive mapping approach and just concatenate your organism of interest reference genome with at least human, with maybe microbes, maybe something else. You can add plants, you can add invertebrates. It's up to you what to include in this database. This competitive mapping, it was advocated in a few papers. So this was one of the papers where I think, yes, so the paper was about like showing that the competitive mapping approach is more accurate. But still, if you check this paper, the competitive mapping here meant that we concatenated elephant reference with human reference, and that's it. So, and of course, yeah, it can be called competitive mapping because two genomes are better than one. But still, what prevents you from adding more references here? Because I mean, obviously, and this is what they claim in this paper is that competitive mapping is more accurate. So it's beneficial for the accuracy of your aDNA analysis. But what prevents you from adding more references in this database? So basically, the approach I'm advocating is okay, if you have a read, even coming from a mammoth task. But in principle, we can double check, we can double check against multiple organisms. So why not to, concatenate your elephant reference with human plus many other organisms, such as plants, microbes, invertebrates, because actually, yeah, I'm not sure how to answer this question. So I'm even if I get a sample, and I was told that it was ancient human, for example. But just to double check, I would think about mapping this read to multiple organisms, because often in this way I learned in my experience that often it's quite non-trivial to say that if you are told that if you were told that this is an ancient human or mammoth sample, often you discover that it's not that simple, and there is actually a mixture of DNA coming from different organisms. So I hope I reiterated the same idea many times. So I apologize if you get annoyed, but that's
Sorry, Nikolay. Do you hear me?
yes, I
I have a question here.
Sure, go ahead.
About decontamination, I wonder, since these samples is like post-mortem, is exposed to deamination, fragmentation, and at basic level, at basic composition. I wonder if there's any also way to decontaminate those fragments.
Yeah, so so this is what I'm talking about. So of course, the gemination it's also a valuable tool for disentangle modern ancient reads. So it can help you to figure out that where the ancient reads, where the modern reads are, but also the contamination I mean and decontamination. What I mean here is more like going beyond ancient DNA applications. So let's say you don't have gemination. You're not working with ancient DNA. Still, even in modern genomics, there can be, you can be given a sample with a mixture of DNA from different organisms. And what to do there? So how do you see that, how can you disentangle microbial reads from mammalian reads? If they're long enough, maybe, but you still need to do this sort of competitive mapping. In my opinion, that's a very straightforward and cheap, computationally quite a cheap way to quickly understand how many organisms are in your sample. So contamination can be understood, in different ways. Of course, ancient DNA people, they call contamination everything which is modern. Often, like they say, well, I don't want modern reads. And then, yes, the gemination or fragmentation, DNA fragmentation is helpful to disentangle modern versus ancient. But then let's say you have multiple ancient organisms present. And of course, if you map everything, let's say you have ancient microbes, ancient plants, and the mammoth DNA. And if you map, this mixture of DNA to the elephant reference, of course, the plants and microbial reads, they potentially can bias everything, can bias variant column, for example. So I hope this answers what you were asking about. So if you, so this is about competitive mapping. I call this approach as ultra competitive mapping. So when I said what prevents you from concatenating this elephant reference genome with multiple organisms. And this is actually was done in one of the recent papers. I think it was published quite recently, 2024, 2025 even. So basically, it's a super simple idea and super low-hanging fruit, in my opinion. So you can, of course, prior to any ancient DNA pipeline, you can just apply Kraken. You can apply this k-mer based analysis to deplete your sample to eliminate somehow reads. Okay, if you're working, if you're interested in only mammalian reads, you can quickly using Kraken deplete your sample by removing microbial reads, for example. That's a quick way to clean your data, and this is essentially what they suggest in this paper. So it's as simple as put Kraken in front of any ancient DNA pipeline. That's it. And they show, so they compare different approaches, and they show that this kind of decontaminating your data with Kraken was more accurate. So the key word here is the accuracy. So I can see a question in the chat. So of course you guys can post your questions in the Zoom chat. Unfortunately, they will disappear after we close the Zoom session. But and also I might not see them immediately. So the best way to ask a question is actually to raise your hand and unmute yourself. But of course, I will be happy to answer your questions in the Zoom chat as well. How fine scale is competitive mapping? If you use very closely related references for competitive mapping, if you think you have too closely related microbes of interest in the sample, will there be an excess of ambiguity? You're right. So basically, if your aim is actually, let's say, people, especially, I'll give you an example from ancient metagenomics, where searching for ancient pathogens such as Yersinia pestis, the plague-causing agent, is a big direction. Lots of labs routinely screen their samples for presence of ancient pathogens. And indeed, so here the challenge is, are we sure that we caught Yersinia pestis reads, or is it Yersinia but something else? Yersinia enterocolytica, Yersinia pseudotuberculosis, something else. So there are lots of species in this Yersinia genus, and yes, so you lack this confidence that you actually found Yersinia pestis and not Yersinia something else. But how to approach this problem? So I would say, so if you have if you concatenate all Yersinia species in the same grand reference genome and let your reads to decide what they're most similar to. Are they most similar to Yersinia pestis or Yersinia enterocolytica, which is an innocent Yersinia? I think that's kind of a more that's the best approach. And of course, you will lose lots of reads because it's probably the majority of reads, they actually will map with the same affinity to both of them. Yes, so in terms of data, in terms of how much data you lose in this way, yes, it is very there will be a lot of waste. You will waste a lot of data because of this ambiguity. Yes, there will be ambiguity. But if the goal of your analysis is actually to claim that there is Yersinia pestis, it's a big deal. It's a big deal to claim that you found Yersinia pestis, because you need to prove that this is pestis and not another Yersinia. And I believe so here you have to waste your data. You have to be on a safe side, and a lot of reads will map with the same affinity to both of them, and maybe a small fraction of reads will map uniquely to Yersinia pestis. while they were given the choice to map to something else, to another Yersinia. But at least if you follow this competitive mapping approach and show that yes, there was a small fraction of reads mapped uniquely to Yersinia pestis, and almost nothing was mapped to Yersinia enterocolytica. And the majority of your reads will map with exactly the same affinity to both of them. Those are non-informative reads. Those you can drop. You can't, you know that you found a genus. You found a genus, so it's clearly Yersinia, but what species you don't know. And you will lose a lot of reads. But if you show that, yes, you ran this competitive mapping approach, you gave your reads the choice to be mapped to Yersinia pestis or Yersinia enterocolytica, and a small fraction of reads still decided to map to Yersinia pestis, despite they were given a choice to map to other Yersinia species. That's, in my eyes, that's the evidence that it is Yersinia pestis. So basically, yeah, I think this is what you asked about here. Yeah, this is the ambiguity you meant, I hope. Yes, let's go further. So I said that this basically plugging in Kraken, to any ancient DNA pipeline. I personally think it's a very good way to decontaminate your data. And again, this is Kraken is a metagenomic tool. I hope you can see that many of the steps that I'm kind of advocating here, they all come from this metagenomic field. And if you're not into metagenomics, just let me tell you a few words about what Kraken is and how it works. So it's basically quite an old tool, published in 2014. I think the first version of Kraken was published 2014. And it's it's kind of a way to avoid alignment. So it's kind of a pseudo aligner. So you're not doing the actual alignment, but what you do here, you pre-process a bunch of reference genomes. So imagine that you have you collected tens of thousands, maybe hundreds of thousands, millions of the reference genomes, and then you can split all of them into k-mers. And you can check what kind of k-mers are more common in mammals and what k-mers are more common in microbes, what k-mers are more common in plants, and so on. So you create this table of k-mer kind of abundances more common to a certain organism. Some of these k-mers can be, species specific, that they are so dramatically more often found in particular species compared to all other species. And this is how you get this species level resolution. But the main idea is that you create this table, a lookup table, and your query reads when you get your ancient DNA sample, you can split all your query reads into k-mers and just check the k-mer composition for each read and see whether these k-mers that are most abundant on this read, they're kind of special for kingdom, for family, genus, species, and so on. So this is how Kraken works. And the k-mers, I hope you guys see it's kind of this a way to get a substring of a nucleotide string. So if you have the nucleotide sequence, you can run this sliding window, and what you read in each frame in this sliding window, this is a k-mer. And then you can sort of count how many times each k-mer is found within each organism and in each query read, and compare. So the idea of this approach is this comparison of your particular k-mer composition in your sample against the database. But otherwise, it's also a database approach, reference-based approach. I can see one more questions in the Zoom chat. Okay, great, thank you. the thing about Kraken is that it's very fast. It's much faster than alignment, and it's quite cheap. So basically, I would say that's such an obvious idea to just run Kraken prior to any aDNA pipeline. That's in my eyes is how it should be done. And again, I would say the majority of people in the field are not doing it this way. But maybe they will. I don't know. It's quite an early days still in terms of this metagenomic mentality. So the Kraken itself can be a little bit tricky for people if you're not used to reading the outputs from Kraken. It looks like this. So you get this strange report with some strange metrics, tax reads, percentage of reads, COV, DAP. What does it mean? And these tax names, unless you're very experienced, evolutionary biologist, which I'm not, you probably get lost very easily here. But there are a few ways to filter Kraken output, and I highly recommend, like the point of this slide, to demonstrate how dramatically you change your your information that you learned from Kraken output when you apply gradually different filters. So for example, you can pick just species. Let's say I'm not interested in this phylum. Let's concentrate on species only. And then suddenly, okay, you get this Loxodonta africana, maybe mammoth DNA, that makes sense. But I also see the strange, other animals like Trichechus manatus, dugon. It's kind of a sea cow. So I'm sure that you're dealing with mammoth DNA, not sea cow DNA. So how can we, get rid of these, organisms that don't make sense? Of course, you can, manually remove them if you don't believe that you have sea cow in your sample. But there are more like intelligent ways. You can apply filtering based on these stats that Kraken provides. So, for example, if you in Kraken Unique and also in Kraken 2 nowadays, there is a way to look at the numbers of unique k-mers. So basically, I said that Kraken is based on this construction of k-mer lookup table and also splitting query rates in the k-mers. So there is a kind of one metric provided by Kraken is a number of unique k-mers covered by your reads. And this is kind of a proxy for breadth of coverage. I'll come to this concept in a second. But I just want you to see that applying this simple filters, like let's select only species with a large breadth of coverage, such as let's say so far it's an arbitrary number 1000, but you get a much cleaner list of organisms. Still, you see this Canis lupus, Homo sapiens, and again this Trichechus manatus, which is a sea cow. Still, it's not good enough. So then you can demand that, well, I want at least 200 reads to be associated with each organism I found. And then you get this crystal clear picture that there is Loxodonta africana, which is probably a mammoth sample. You get some human contamination and you have some bacterial contamination, just 3 organisms. And this was a simulated sample. So actually, this is a ground truth. So I know that I simulated in my sample these 3 organisms. So basically filtering, it critically changes what you learn from Kraken output. So basically you never, you never take Kraken output as is. You should always filter. That's a take-home message. I see another question in the chat. Considering that ancient DNA is highly fragmented and chemically damaged, how does Kraken handle the classification of ultra-short and error-prone k-mers without increasing false positive taxonomic assignment? So that's what when this tool was developed, it was specifically tested. This sensitivity versus specificity, and then so it's a pretty accurate tool. Like short, long story short, my short answer is that I'm pretty sure that Kraken is quite specific and quite accurate. So it was not tested on ancient DNA. So all these tests were done on modern reads. However, Kraken is widely used in aDNA analysis as well. Just because the idea of this K-mer approach is that you split your query reads into K-mers, and then all these K-mers, they vote for a particular organism. So what you typically see is that there will be reads, that suggest, there will be K-mers that suggest that your read belongs to human, and then there will be a group of reads suggesting that your read belongs to cow, and then there will be reads suggesting that your read belongs to maybe microbe. So Kraken does this majority voting. So basically, what is important is that the majority of reads should suggest a single organism. And then, yes, you can have damaged reads, but then it will be, just a few K-mers. Still, the majority of the K-mers coming from your read, they will vote for correct organism. So basically what I mean is that this domination and damage, it doesn't affect the accuracy of Kraken analysis much. So of course it affects a little, but not dramatically to my experience. So domination is kind of a minor problem for Kraken. And then now about fragmentation. So Kraken, well, at least I would say it's a gold standard. You never analyze reads shorter than 30 base pairs, because 30 base pairs is like this kind of specificity threshold, the minimal specificity threshold. So below that threshold, there is kind of ambiguity. You can't be sure that this read belongs to that organism and not another one. So you lose the specificity, that's why Kraken, I think by default, Kraken uses 30 as a minimal read length. So if you, and of course for ancient DNA people, that might be frustrating because many reads will be, 20 reads or 18, 20 nucleotides or 18 or 10 nucleotides. But basically, it's better not to work with those reads. And yes, you will lose a lot of reads, but are they useful? Are they informative? No, it turns out that you can keep these reads, but it's risky. So basically, you better not to keep them. And Kraken does, in my opinion, the correct thing by dropping those reads.
Sorry,
yes.
Nikolay, we have one more question about Kraken.
Yes.
Chat.
Yeah. There was so please go ahead. Swati is asking, I wonder if the K-mer size can be set to a value below 30 or something, then probably ancient reads can be taken. No. Basically, no, I wouldn't recommend. To decrease this, default. I think by default, Kraken indeed uses 31 or 35, and I would not recommend to decrease this threshold for ancient DNA analysis, simply because you lose more than you gain. Yes, potentially you can rescue lots of valuable short reads. On the other hand, you lose more. You sacrifice the accuracy of your analysis. So if you go below 31, do it at your own risk. So you should realize that you're sacrificing the accuracy of your analysis, and then you can decide for yourself whether it's okay to sacrifice the accuracy. Yes, so these filters, the filters that I just mentioned here, the K-mers, unique K-mers, and a number of reads, they have something to do with these known concepts, breadth and depth of coverage. I think people who do, single genome approach, and if you do this alignment of your reads to the reference, you're familiar with these concepts, the difference between depth of coverage, a metric which is important for people interested in variant calling, so they would like to have a decent amount of reads supporting each, minor allele, But for people doing metagenomics, it turns out that the breadth of coverage is a more important metric than the depth of coverage. It turns out that often when you do metagenomic analysis, you might have, if you look at the alignments in IGV, you might have piles of reads aligned in just a few locations, a few loci. And this probably means, one interpretation of this is that these reads are actually not coming from the organism that you are visualizing alignments from. So they actually came from another organism, but they're misaligned here now. And that's why you have these piles. So let's say here, so here the situation in this slide is not that bad, but if you had, empty here in between and just two piles of reads, that would be an indication or suggestion that probably these reads aligned here, they're actually coming from another organism. And just this particular region is conserved between that organism, whose reference genome you're using, and another organism who was the source of those reads. So I hope this is clear. And I see another question in the chat. Thank you for asking the question, by the way. And Kraken, as I said, it's important to filter Kraken output. Without filtering, it's kind of quite useless, just the Kraken output itself. So you should always filter and only after that start interpreting. And the Kraken basically can implement both depth of coverage and breadth of coverage. You should have like a decent number of reads. You shouldn't, judge that you found a certain organism based on 5 reads. Basically, that's a depth of coverage. The number of reads is kind of proportional to the depth of coverage. But the certainty and specificity of the organism you found, it's provided by the breadth of coverage. So basically, people working with single organism doing this animal ancient DNA research, working mostly with single organism, you probably you don't look a lot at this breadth of coverage, probably, but for you're working basically mostly with depth of coverage because it's important for PopGen, for variant calling, and so on. But for metagenomic people, if you happen to work with ancient metagenomes, breadth of coverage is actually much more informative metric compared to depth of coverage, and you usually should filter your Kraken output here with respect to both depth of coverage and breadth of coverage. So that's a picture from the ANITA publication from our paper where we demonstrate that if you sort of apply reasonable thresholds on breadth of coverage and depth of coverage, you get very reliable list of organisms through Kraken. And Kraken is, so I said, it's a reference-based approach. It's very database dependent. I would say, so I reiterated this many times. I hope you know by now what I mean. So basically, when I concatenate multiple organisms into grand reference genome, I'm increasing my database size. This is what I call database. And Kraken is very dependent on the size of the database. So basically, when I say dependent, it means Kraken is more accurate for large databases. So database construction is an absolutely central thing about Kraken. And in our AMITA publication, we actually demonstrate the importance of the size of the database. So we actually used some simulated ground truths, some simulated reads, and we tried to reconstruct this simulated ground truths by running Kraken on different databases. So we compared, so here on the x-axis, it's database size. So we ran Kraken on a number of databases. And every time we compared the accuracy, the overlap between reconstructed, example, it was microbial composition, reconstructed microbial composition with ground truths, simulated microbial composition, and so the y-axis, it's kind of the accuracy of reconstruction of the ground truth. And what we demonstrate is that yes, larger databases, they are simply more accurate. That's why I'm saying that, like, the more the more organisms you put here, the more accurate you are. Yeah, I hope you see the point here, the message. And then if you happen to work with environmental ancient DNA, the situation is even more extreme there. Because so basically, if you have a mammoth task, yes, you may assume that there are a few more organisms, but you at least have some idea what that it can be, human DNA, maybe microbial DNA, but at least you know the host. At least you know the host. Like the host was, it was a mammoth task. So your analysis is more like focused around mammoth DNA. But if you're working with environmental sample, you have no idea what is inside. So in this case, you actually you should do even more extreme competitive mapping. So you should include in your database. You should include all mammals, all non-mammalian vertebrates, plants, invertebrates, microbes. So the databases become gigantic. And this is simply because you have a mixture of reads and you want to disentangle them. You want all these reads, they should find their respective references in their respective kingdoms and their respective databases. That's why we need this gigantic database. There was one more question in the chat. Danny is asking, is there a point after which you get diminishing returns in terms of database size? I imagine it varies significantly by taxon and DNA quality, but perhaps not. Yeah, so it's something to test, Danny. But basically, in this particular example, what we saw is that since the ground truth, simulated ground truth, was microbial DNA plus human. So in this particular exercise, in this publication, Genome Biology 2023, the ground truth was mostly microbial because it was ancient microbial DNA simulated. That's why there was some sort of saturation here because like microbial NCBI-NT was almost identical to the full NCBI-NT in terms of that adding, eukaryotic DNA to the database did not improve the accuracy of ground truth reconstruction because the ground truth was microbial. So adding eukaryotes did not improve this. That's why there was this saturation. But otherwise, I hope you can see that just complete NCBI-RefSeq was very inaccurate. And you should do better than this. You should, include more. Your database should be diverse. Ideally, it should include, all organisms. Yeah, preferably, if you want to be completely unbiased and open-minded, all organisms across tree of life. But otherwise, I think this is what you were asking about, the way I understood your question, Danny. Yes, and then the database size. The database size is a problem because large databases, they require a lot of RAM. And here I'm just giving you, so this here, it's a size of the FASTA file, basically in gigabytes. But here you see the RAM, which is needed by Kraken Unique if you want to do, if you just create a database around mammals only. So you will go above 1 terabyte of RAM. If you want to run GTDB, which is a microbial database, and want to run your ancient metagenomic samples against GTDB, well, you need more than 2 gigabytes of RAM. And I realized that, and this is a combined, this is how much memory you will need if you put all of these guys together in one database. And I realized that very few people in this audience, they have this luxury to have access to 4 terabytes of RAM. That's why, yes, so here there is a bottleneck. Indeed, amount of RAM is a bottleneck. But on the other hand, if you don't have enough RAM, then you sacrifice the accuracy of your analysis. So you have to choose. So if you don't have, if you say, well, I have only 500 gigabytes of RAM, that's it. That's the maximum I can afford. Can I do analysis? Yes, you can, but it will be inaccurate. So you have to realize that you're sacrificing the accuracy of your analysis. You have to clearly realize this. And yes, one potential, workaround is to do chunking of your database. So this is what Copenhagen, I think, School of Ancient DNA, they're using a lot. They kind of providing these databases individually. So their query reads, they exposed against this database individual, and then there is a clear, there's a smart way to overlap. So you can actually run your classification against these databases separately, but then you have to overlap. You have to overlap somehow the outputs and remove these redundant reads that map to more than one database. For example, that's a potential workaround. Yes, so let's move on. Decontaminating references. So that's that's the essence of the GeneX method that we developed recently, and I'll try to explain another problem in ancient DNA analysis. So I was talking a lot about contamination of the reads. I said that we actually work with this horrible mixture of reads, and maybe some of them are interesting for you guys, but some of them you can call them contaminant. So maybe you don't want microbial reads. So microbial reads are contaminants for you, maybe. But there can be another type of contamination, not only in your sample, but it turns out that there can be contamination in the reference genome. So let me explain it slowly. So I said often we are not sure what kind of organism this read belongs to. If I was told that it's a human read, who knows? If I'm open-minded, I can assume multiple organisms. Let's double check. Let's be on a safe side. Yes, I suspect it is human. It is a human read, but let's double check. So if I want to be open-minded, I go this competitive mapping approach. I map my read against multiple reference genomes, but are they okay? So here, yeah, maybe I will figure out that my read maps with the highest affinity to my human reference. Does it mean that I found a human in my sample? Not always. So the answer is that not always. And yes, so let me check the question first in the chat. So what he's asking, I have a technical question related to the GeneX method. I'm using, okay, excellent, thank you. Maybe do you mind, Swati, if I just explain this method and then I'll answer your question. Would it be okay? Because currently, if I start answering your question, people don't know what this GeneX. They don't know how this GeneX works. Maybe it's too early to answer your question, but I promise I'll answer it like in a few minutes. So yes, the question I'm asking here, are these reference genomes okay to use? Is it reliable if I demonstrate that this read maps to a human reference? So it turns out not always, because there are multiple examples that within human reference or elephant reference, you can have chunks of microbial inserts. And other way around, in microbial references, you can have eukaryotic regions like human contamination, etc. And I'm not actually exaggerating. There are tons of literature showing that yes, there is a problem with contamination of the reference genomes. And then if you guys, if you're running deep sequencing experiment on a mammoth task, maybe. So you have this deeply sequenced mammoth. Apologies for using mammoth as an example, but by mammoth I mean any other organism, sheep, reindeer. Pick your favorite organism. But I'm here kind of using mammoth as like a general ancient animal. Might be interesting for many of you. If you have a deep sequenced mammoth sample, Yeah, even if there will be a few inserts from microbes, but we can live with it. So it's not a big deal actually, because you have millions of reads, and even there will be some parts of elephant genome contaminated. It's still okay. It doesn't change the overall message from your analysis, because it's probably it's tiny fraction. So let's say that this particular elephant reference is not badly contaminated. Yes, there will be, I believe that there will be a few inserts, but probably it's a small fraction of reference genome which is contaminated. So it's okay. We can proceed with that. The problem here is that, so there is one particular area where it is a big deal that tiny fraction of reference genome might be contaminated with microbes, and in this area is actually ancient environmental DNA. Because I'll explain why. But here, the peculiarity of ancient environmental DNA is that typically there is no host organism, so you don't know what is inside. So you have to be extremely open-minded, because you can't assume a single organism. And then the amount of DNA per organism is so low. I mean, we in ancient DNA, we know that yes, amount of DNA is never enough for us. But imagine now the total amount of DNA you sequence, it is low. But if you do assignment of certain reads to an organism, it will be, you're down basically to 20 or 100 reads. So actually detect these animals based on just a few reads. Think about it. So this statement that you found a mammoth in the soil is based on how many reads? 20, 100, maybe 1,000. And then, of course, if you're coming from PopGen, people might ask, how tall do you do analysis with 100 reads or 1,000 reads? So you can't do any analysis. Well, here with environmental ancient DNA, we are not actually into variant calling and this inference, phylogenetic inference, not always, but for this area is enough to say that I found or not found. So this 20 or 100 reads, they're actually enough to answer this question that I found or not found. Yes or no answer, basically. That's what the amount of DNA allows us to answer at least presence or absence question. And so that's a hot area. I hope you guys know about these high-profile papers in Nature and Science. And yes, so it looks spectacular. So if you look, for example, this heat map, it's all these animals found in Greenlandic samples. But if you check the supplementary material and check what is the number of reads supporting each of these, dark region on this heat map, and you go actually to supplementary and check what is the amount of reads, that's the amount. So actually we claim that we found certain organism based on a couple of hundred reads, maybe at best a couple of thousand reads. Why is it problematic? Well, it turns out that here, these high quality references, they're particularly important, because you better make sure that this a few hundred reads, they're actually not coming from those very, very few regions that are potentially exogenous. Imagine that, so here maybe, 0.1% of elephant reference is exogenous. Are you sure that these reads, coming from this couple of hundred, couple of thousands reads, are they coming from those very few exogenous regions, or are they coming from the endogenous regions? That's a good question to ask. This is exactly the point of this method, the GeneX method. So we did a super simple thing. So we just, we took one of the largest microbial databases, which is GTDB, and we just chopped all the microbial references into pseudoreads. So we kind of produce these pseudoreads, and we aligned them to all NCBI eukaryotic references. And also phylonorway, so one data set that was used in this studies, a Greenlandic paper, a 2 million year old paper. So they use this phylonorway data set. So it's a collection of plants. It's a 1,300 plant reference genomes. So we just wanted to create a map, or a map of where this exogenous, if there are exogenous regions, where they start and where they stop. We believe that this is a valuable resource for the community to potentially mask this region. So if you guys suspecting that your reference is not clean enough, and you're operating with very few reads, maybe you're doing an environmental ancient DNA research. So I think for you guys it would be very valuable to actually mask, to use our map, these bed files with coordinates of exogenous regions, and mask the reference genome of your favorite organism. And this is what we are suggesting basically in our GigaScience paper. So we are taking this original reference, we detect where this microbial pseudoreads map, and we just use this coordinates for masking. So it's not a reindeer reference anymore, but masked reindeer. In this way, you're on the safe side. So you remove this potentially risky regions. So again, it's not, there can be multiple explanations why this microbial reads map here. But when you do, this risky research, and you're claiming that you detected mammoth from a soil sample, or another exotic mammal from a soil sample, you better be on the safe side. You better actually, make sure that these reads, these a few hundred reads, they're not originating from suspicious regions in your reference genome. So we we ranked all, so we processed something like 4,000 NCBI RefSec reference. So maybe your reference genome of your favorite organism was already screened in our paper. So we provide these coordinates for around 4,000 references. And we ranked all this mammalian and invertebrate and plant organisms by their percentage of microbial-like sequences. And overall, the picture is not bad. So since the majority of our reference genomes, they were NCBI references, RefSec reference, so they're quite well curated. And so what we found overall is that in the worst case, like this antelope, for example, it had maybe 1.5% of contaminated, or what we call microbial-like sequences, but it's not, it's probably not a big deal. However, in this particular phylonorway plants data set, we found something very interesting. So these numbers on the x-axis, those are the percentages. So according to our analysis, you can have up to 70% of this reference genome which looks suspiciously exogenous, that doesn't look like a plant. Unfortunately, and that's a good question to ask, what is going on. So basically this phylonorway data set, it was standing out clearly in terms of microbial-like breadth of coverage compared to other groups of organisms. So we also took this phylonorway reference genomes, and we split them into what we predict are endogenous regions and what we predict are exogenous regions. And we computed the k-mer distances with MeSH, pairwise k-mer distance between all the references from the phylonorway dataset. And here, I think it's pretty clear that these microbial-like regions, they cluster very distinctly from what we predict plant-like regions. And this simply shows that there is a difference in k-mer composition. So there is something dramatically different in the k-mer composition of these regions compared to those regions. So we also did kind of a lot of follow-up. We were checking this Hyperis vulgaris heat, which was kind of number two most microbial-like or least endogenous reference genome. And also, yes, so all these Kraken screens show that although there are contigs that look like plant contigs, but the majority were actually bacterial. And we also did this kind of phylogeny type of analysis. So since we can compute pairwise distances, k-mer distances between different regions of Hyperis vulgaris. So we also we tried to project the Hyperis vulgaris endogenous regions. So basically, these nucleotide sequences coming from endogenous Hyperis vulgaris regions and microbial-like Hyperis vulgaris regions. We tried to project these two types of nucleotide sequences into this dendrogram containing bacterial ref-seq and plant ref-seq. And what we saw is that what we predict microbial-like Hyperis vulgaris regions, they were clustering together with bacterial ref-seq regions, and what we predicted were endogenous Hyperis vulgaris regions. They were clustering together with plant ref-seq regions, again suggesting that probably those two types of sequences within Hyperis vulgaris are quite different in terms of k-mer composition. Also, these heats Hyperis, they were mentioned actually in these two papers, Nature papers, and we did a quick lookup. So we just took one sample from this Arctic study, and so it had in this sample Hyperis vulgaris was reported together with a few mammals. So we took just these reads that were publicly available. So we just took these reads and aligned these reads against Hyperis vulgaris reference from the Phylonorway dataset plus a few mammalian references. Doesn't matter what mammalian reference. It just we simply wanted to, we wanted this mammalian reference to attract mammalian reads. So we thought that okay, this is needed to like disentangle mammalian from plant reads. And here, what was aligned to Hyperis vulgaris is probably plant. What we believe is plant. And let's see whether these reads aligned to Hyperis vulgaris, they actually fall within our annotated, microbial-like regions or endogenous regions. And the thing about Hyperis vulgaris is that approximately what our method, GeneX method, predicts, approximately half of its reference is actually free from microbial-like regions. So basically, these reads aligned to Hyperis vulgaris. They have a choice. They can align here to this red endogenous regions, or they can align to those microbial-like regions. And let's do some enrichment analysis. Let's check how often they align to the endogenous regions and how many times they align to the exogenous region. It looks like in the vast majority of reads, they actually aligned to this, what we predicted were microbial-like regions. So that's a little bit a danger in this type of analysis that please make sure, please prove, if you find, an animal DNA in your sample, in your environmental ancient DNA sample, please show that it's actually not a microbe you found. Because these microbial reads, they very happily can map to your mammalian references for different reasons. They can be too short and too non-specific. So microbial reads, even if you're talking about modern microbial reads, they can, they can happily map short enough reads. Microbial reads can happily map to a mammalian reference. But also, in addition, you might have, these microbial inserts in your reference genome. So if you're fishing out, animal DNA from a soil using references of poor quality, you're at risk to to actually detect something else than the organism you're thinking about. Yeah, so let me skip this. So it's basically the end. So I prepared a lot of slides, and I hope it's been interesting for you guys so far. And yeah, I'm about to stop because we need some time for discussion. But these steps like authentication, I think it's not new for you. I think everyone knows the map damage and this what we are checking, this enrichment of transition mutations at the ends of the reads, and this fragmentation should be that's two basically evidences that you're dealing with ancient DNA. A tool that I would like to emphasize here, what I think is better than map damage actually, that I'm a big fan of this tool, PMD tools, because often I personally deal with user or UDG treated samples. So for me, it's actually quite important to still check the domination pattern, and PMD tools can use the CPG sites to check domination, which kind of escapes user treatment. So CPG sites, they kind of escape in this treatment for some unclear for me reason. Yeah, so that's super important to still see the domination even on treated samples. Second thing that I particularly appreciate about PMD tools is that it provides, it's to my knowledge, it's the only tool that gives a per read prediction, ancient, non-ancient. So it gives you some sort of score, post-mortem damage score per read. And this is a little bit spectacular because actually map damage, it operates, so it needs like a couple of hundred reads, maybe a couple of thousand reads to make this beautiful domination profile. While PMD tools, in theory, it can give you yes or no, ancient, non-ancient status per read. And this is beautiful, in my opinion, especially for, again, for environmental ancient DNA people who deal with just very limited number of reads. There is no way you can run map damage on 20 reads. But with PMD tools, there is a chance that all these 20 reads, they get high PMD scores. That's, well, it's a weak, but still some evidence that suggests that probably those reads are probably ancient, even though it's 20 reads, but there is kind of some indication that they might be ancient. Also, what I'm personally developing in my group that I hope we can publish soon is this very old idea that I was formulated like back in 2018 and never had time to wrap it up as a proper publication. So it's basically so what the original idea was is to try to use CNN convolutional neural network architecture for basically learning modern sequences versus ancient sequences. So it's a machine learning approach. Let's nowadays we have tons of modern sequences and tons of ancient sequences. Let's just train a machine learning classifier such as a convolutional neural network that can learn k-mer composition, which is more typical for modern reads and more typical for ancient reads. And yeah, so some preliminary analysis was done by me like many years ago, and there is a way actually to interpret this neural network and check so what are the most informative nucleotides. Even though people say that machine learning is kind of a black box, I disagree. So you can always check, look inside and control all the parameters, all the gradients in the neural network. So basically, there is a way to interpret your prediction. So if your neural network can discriminate between modern and ancient sequence, you can check what kind of nucleotides are most predictive for this purpose. And it turns out, like in my preliminary experiment, it turns out that these ends of the reads were most informative, which makes sense because domination happens at the ends of the reads. But now, happily, Daniel Rivas is actually looking into this in depth, and hopefully we can wrap it up as a nice tool, which can be complementary to PMD tools. Basically, it will be a tool that can give you per read prediction, so ancient or non-ancient, and potentially, like another idea is that potentially we can detect something else than just domination. So the idea that I had for a very long time in mind is that maybe there is something else than domination that is more typical for ancient sequences. So let me just quickly check the chart. Yes, not sure if widely used, but I have also used a tool called Authentic. Yes, it's yeah, yeah. I know. It's from the Leipzig group. Yeah, I forgot the name of the first author, but yeah, I know this tool. Absolutely. It's not, yeah, I have limited experience, unfortunately, with this tool. But so let's maybe move to the questions. So I think I'll stop here. Let's revisit the question that I promised to answer about the GeneX. So Swati is asking, I have a technical question related to the GeneX method. I'm using it at the moment. In fact, I started using it yesterday and I'm waiting for the results from it for a custom database. I'm just wondering if the output from it would be in the form of a bat file with coordinates of microbial-like regions, and the masking of those regions will need to be done manually later, or if the method creates a final Bow-Type 2 database with those regions masked. Unfortunately, it's only the bat file. So there is a very easy way to mask any FASTA file with sorry, what is it called? Bat tools. I think it's bat tools. You can use for quick masking of a FASTA file. If you have the bad coordinates, so if you so this GeneX method, it will give you the coordinates of regions that you probably would like to mask. And then if you have this bad file and you have your FASTA to be masked, basically just a few minutes of running bat tools. So there is just one command line which I can share if you want. But I think in the supplementary, yes, in the supplementary for this GeneX publication in GigaScience, we actually provide the command lines how to mask the FASTA file. But yes, so the short answer is that GeneX gives only the bat file, not the masked FASTA. And you have to mask it yourself, but it's so easy to do with bat tools that I hope it's not a big deal. Okay, thank you so much. Oh, okay, guys, apologize. It was so many slides, and yeah, I can talk more. Unfortunately, we need to stop somewhere because I can see that I'm running out of time. And please go ahead with your questions. So maybe we can spend a few minutes. But otherwise, you were very good asking your questions during the presentation. So I hope many of them are already clear. Thanks everyone for your attention.
Thanks so much, Nikolay. That was great. Given that you had only 2 hours, a lot of information and a lot of useful tools. If we have a couple minutes for questions, and if anybody wants to unmute themselves or write them in the chat, I can read them out. If not, I can also quickly ask one myself. So a lot of people probably in this Zoom at the moment or working in the ancient, like in the art community, will work with a lot of like non-model species. For which, I mean, you touched a lot on the reference genome aspect, but either they're not very high quality or they're using reference genes from that are not from the species that they're target, but closely related species. Like all these approaches and kind of pipelines you touched on today, you think these are could be more useful in such cases, or is it still could be kind of tricky since sometimes some situations where you don't have the ideal toolkit of your study species to include?
Yeah, so the way I understood your question, you're asking about this biological divergence between, so maybe you're using, elephant as a proxy, but in fact you're dealing with mammoth reeds. So this kind of biological divergence is what you're asking about, like this discrepancy between the reference and the query reads can be not due to the quality, but just the biological divergence, and maybe it's suboptimal to use elephant for analysis of mammoth DNA. Sorry if I misunderstood your question, but I hope you were asking about this.
Yes, also including that in addition to, I mean, there are different problems. You can have bad quality reference genes, you can have
Yeah, so I
contamination, as you mentioned, missingness, yeah.
Yeah, so I highly recommend if you have this evolutionary divergence, so you're prone to this reference bias. So you basically the risk is that you will select reads that resemble the reference the most. But what if so you're kind of you're making your ancient organism more similar artificially, more similar to the modern reference. So you have to realize this that maybe evolutionary divergent reads can be dropped if they will simply have too many mismatches. So you have to realize that by doing whatever mapping, whatever reference-based approach, you are selecting reads that resemble the modern references, and this is what's called reference bias, and it's a big problem. So basically, uh, there are suggestions in this paper what to do with that. But maybe like the most straightforward answer is not to use very short reads. And of course, it's very frustrating for ancient DNA people because we would like to keep as many short reads as possible. But again, you have to realize that yes, you rescue a lot of valuable data, but you're sacrificing the accuracy of your analysis in different ways. So you're either prone to contamination, so by rescuing short reads, you are enriching your sample by contaminant reads. First of all, second, you also what they show in this paper, like you're also prone to reference bias and. Yeah, so basically, but there is what you're asking about this evolution divergence is a more difficult problem. It's something that there is no there are approaches, there are different suggestions, but not straightforward.
And I guess this aligns with another question we have in the chat from
Oh, so
Camilla.
many questions. Sorry.
But maybe I think we kind of addressed.
The guys are saying that yes, thank you, and they
There's
have to go. Fit well. Okay, here there was a Kavia is asking probably a very specific question. I'm working on some extinct species and using competitive mapping with the closest modern reference I can find and human. However, I'm wondering how I can make the mapping more accurate, especially to avoid losing reads that don't fit well. Well, yeah. That I think we partly already discussed this just a few seconds ago. Yeah, this reads that don't fit well. It's the good question is why, and it can be biological reasons due to this evolutionary divergence, or it can be technical reasons due to contamination or degradation. But in any case, I would say those reads are riskier in general. Yes, so that's a compromise. So you maybe would like to keep them, but you have to realize that it's too risky. And as I said, in ancient DNA, well, compared to like modern genomics, what is shocking is that lots of statements, like amazing statements, are done based on very little evidence. You have to realize that you need to probably do much more work to still use these reads that don't fit well. So you have to convince the community that you analyze these reads correctly and you made these inferences accurately. So, but it's again, of course, it's a hard question what to do with them.
Thanks so much again, Nikolay. I think we can stop here. And if anybody has more questions, I assume they can shoot you an email and contact you. But yeah, thanks again so much for your presentation today and your time to answer all these questions. And I highly encourage everyone else, if you would like to continue participating in the series, to keep an eye out. We'll send out soon the registration form for the upcoming workshop and start advertising those that will follow as well. So yeah, I guess we can give thank you again, Nikolay, and hope to see everybody again soon.
Thank you, everyone. Cheers. Have a nice day. Bye-bye. Take care.